In this activity, we're going to model mitosis using simple materials so you can physically see how chromosomes move and separate during each stage of cell division. Even though this is a model, the sequence of events you'll observe matches what happens inside real plant cells. Most of the time, DNA exists as loose chromatin and can't be seen as individual chromosomes. During mitosis, chromosomes condense and become visible, allowing us to follow their movement. This model helps make that process visible and easier to understand. I've taken photos of each step I do with the students, but feel free to do it yourself, which helps the process stick more. I provided the materials list if you want to join along. Before we begin, answer this question. Why is it important that mitosis produces two genetically identical daughter cells? Think about the purpose of mitosis, such as in growth and repair. Pause the video and write your prediction in your lab notebook. Then continue. Here I am modeling Interphase. The string represents the nuclear envelope, and each paper strip represents an unreplicated chromosome. We have a red and blue strip of paper, which signifies a diploid chromosome, which is denoted as 2N. The red paper is the chromosome contributed by mom, and the blue one is the chromosome contributed by dad. During interphase, DNA exists as chromatin, where DNA and proteins are more relaxed and not visible to the naked eye. If I was going to make the paper model more realistic for interphase, it would be super long and skinny, coiled in the nucleus. Keep in mind that plants have a diploid number, 2N, ranging between 10 and 100, although there are some, like slender goldenweed, that has 2 pairs, and the highest is the adder's tongue fern, with 720 pairs. At this stage, DNA has not yet been copied. During S phase of interphase, each chromosome is replicated. Each pair of same-colored strips represents sister chromatids joined at the centromere, shown by the paperclip. The centromere is a constricted region on a chromosome that acts as an attachment point for spindle fibers during cell division. In prophase, the nuclear envelope breaks down and chromosomes condense. Spindle fibers begin attaching at the centromeres. This is why the string is removed and your fingers act as spindle fibers. Spindle fibers are microtubules made of protein filaments that help align the chromosomes and pull them apart. They attach to kinetochores, which are protein structures on centromeres. In metaphase, chromosomes line up along the metaphase plate. The centromeres are all aligned at the cell's equator, preparing for separation. In anaphase, the centromeres split and spindle fibers pull sister chromatids apart. Each chromatid is now considered a daughter chromosome. In telophase, nuclear envelopes reform around each set of chromosomes. The chromosomes begin to relax back into chromatin. In plant cells, cytokinesis occurs by forming a cell plate. Folding the paper models the new wall forming between the two daughter cells. During telophase, the Golgi apparatus sends vesicles filled with cell wall materials to the center of the cell, guided by a microtubule structure called the phragmoplast. These vesicles fuse together to form the cell plate. This plate grows from the center outward until it fuses with the original cell wall, successfully splitting the parent cell into 2 new daughter cells. At the end of mitosis and cytokinesis, 2 genetically identical daughter cells are produced, each containing a complete set of chromosomes. Although these cells are considered identical in terms of their DNA, some differences can still occur. First, the daughter cells are smaller than the original mother cell because the cytoplasm has been divided between them. As the cytoplasm splits, the organelles within it are also distributed between the 2 cells, and this distribution may not be perfectly equal. In cells that contain chloroplasts, for example, it is possible that one daughter cell may receive fewer chloroplasts or, in rare cases, none at all. When this happens in leaf tissue, it can result in white or pale patches where photosynthesis cannot occur. In addition, although DNA replication is highly accurate, occasional mutations can occur during the copying process. The likelihood of mutations can be influenced by factors such as the age of the plant, environmental stresses, and the type of cell undergoing division. Compare and contrast the chromosome sets in each daughter cell. How are they the same? How are they different from the original cell before DNA replication? Pause the video to answer the questions. This model shows how carefully regulated mitosis is. Every movement ensures that each new cell receives the same genetic information, which is essential for plant growth, development, and repair.